Welcome back to Bio 111. We're looking at Chapter 15, Section 2. We're going to be looking at prokaryotic transcription. Right, some general steps of transcription. We're going to have the initiation, so that's what starts everything off. It initiates. There's a promoter, which is a piece of DNA upstream of the DNA that indicates where the RNA polymerase should bind and start the process. Elongation. RNA polymerase adds complementary nucleotides A, U, C, G to make the mRNA. So what's going on is it's looking at the DNA and it is bringing the complementary nucleotides to make the complementary RNA strand that matches the DNA template. Again, notice that we don't have a T. We don't have thymine. Thymine in RNA is replaced with uracil. And termination, that is the completion of the synthesis or the transcription of mRNA. Now, if you recall from prokaryotes, there's no nuclear membrane, right, in prokaryotes. So there's no separation between transcription and translation. In eukaryotes, you'll see transcription happens inside the nucleus. Translation happens outside of the cytosol. There's no separation there, and so the two things can happen simultaneously. A ribosome will grab onto the growing mRNA and start translation. So you can have DNA with polymerase making RNA, and that same RNA having a ribosome attached to it making protein. All right, so what we're looking at here is just a cartoon of RNA polymerase taking or opening up a strand of DNA and then making this complementary mRNA strand. Just pairing up complementary base pairs. So the delta subunit of prokaryotic RNA polymerase recognizes consensus sequences found in the promoter region upstream of the transcription start site. The delta subunit dissociates from polymerase after transcription has initiated. All right, so you've got a region of the DNA that will bind polymerase, chemically bind it, and that's what initiates transcription. Remember, this is all just really complex chemistry. Complex in the sense there's a lot of atoms going on here. You got to think about it in terms of binding. Polymerase is a large molecule. It's a protein, a large protein. DNA is just another large molecule. And what causes the polymerase to attach to the DNA? They have some chemical affinity for each other. They'll bind to each other and begin the process. All right. A lot of times we like to think of this. I think genetics and when it comes to biology, it's being very magical. It's just chemistry. All right. All right, elongation. So what's going on with elongation? They're just continuing to add on to that mRNA strand, adding complementary bases to it. It synthesizes the mRNA from the 5' end to the 3' end, and unwinds unwinds and rewinds the DNA as it is read. So basically, it's opening it up and then closing it back. Elongation occurs at the rate of 40 nucleotides per second. All right, so termination liberates the mRNA strand. That means it ejects it, removes it from the DNA. Occurs by Rho protein interaction, formation of an mRNA hairpin. By hairpin, it loops back, the mRNA loops back onto itself and forms a structure that looks a lot like a hairpin, like a bobby pin. Two types of termination signals: Rho dependent, controlled by the Rho protein, row independent, controlled by sequences in the DNA strand. All right, so in prokaryotes, this doesn't have to happen. The process we just looked at doesn't have to happen one at a time. Multiple polymerases can be translating a particular gene into mRNA at the same time. As we see on this cartoon, we're seeing multiple examples of RNA being translated or transcribed on a single strand of DNA, and then on top of that, we see multiple ribosomes translating that mRNA into proteins. That drastically increases the amount of protein that can be generated from a single strand of DNA. All right, so that's it for section 2. Come back in for the next section, section 3, in the next video.